The fossil record is built on the durable. Dinosaur bones, teeth, and thick shells are the materials capable of withstanding millions of years of burial in the earth. Delicate biological tissues, like organs, gills, and developing embryos, are almost entirely consumed by bacterial decay within days of an animal dying. The Isle of Wight in southern England is a primary source for these kinds of resilient, hard-bone dinosaur fossils, but a 125-million-year-old freshwater shellfish discovered there appeared ordinary on the outside. When scientists placed this shell under powerful microscopes, they found a biological anomaly. The mother's soft gill tissues were completely fossilized, and resting within them were tiny, unborn larvae. Normally, the intense heat and geological weight of the Earth would flatten or dissolve these microscopic cells long before they could turn to stone. These embryos only exist today because of a specific sequence of geological events that happened immediately after the mother died. 125 million years ago, the moment this mother shellfish died with her young inside, a biological countdown began. Scavenging bacteria immediately invade the interior of the shell to begin breaking down the soft internal reproductive structures. But in this rare instance, the environment intervened. A sudden influx of fine sediment and mineral-rich water completely flooded the interior of the shell. This thick mud sealed the interior, creating an oxygen-deprived environment that stalled the bacterial decay. This diagram shows what happened at the microscopic level. Mineral-rich water seeped directly into the delicate biological cell membranes before they had a chance to rot. Molecule by molecule, ancient minerals dissolved the original biological material and replaced it with stone. This rapid replacement process created a perfect stone mold of the fragile tissues just hours before they would have disappeared forever. This prehistoric nursery only exists today because the chemical process of mineralization won the race against biological decay. In the mid-1800s, researchers frequently found a dark, unidentifiable substance inside fossilized shells. They called this material molluscite, but the microscopes of the Victorian era couldn't reveal its actual composition. The Isle of Wight shell contains that same dark substance, allowing researchers to study it with modern imaging technology. Scans of this fossil show that molluscite is actually the mineral-replaced soft tissue and reproductive organs of the mother shellfish. This realization means that evidence of ancient soft tissue has been sitting on museum shelves for over a century, unrecognized until now. The preserved mother belongs to an ancient species of freshwater shellfish called Margaritifera valdensis. This data visualization charts the physical progression of the offspring found inside her gills. It maps the development from simple, single-celled embryo structures on the left to fully developed shelled larvae on the right. This shell provides the oldest physical record of maternal care in the history of freshwater mussels. Rather than releasing eggs into the water to face predators alone, this mother actively protected her young inside her own body. That same physical protection is visible today in her living relatives, the modern freshwater pearl mussels. A lucky geochemical accident was required to preserve this specific shell, but the biological strategy of maternal care was effective enough to survive the extinction of the dinosaurs and continues to thrive today.